right, let's dive deep, and I mean really deep, into the code that writes the story of every single living thing on this planet. We're talking about the blueprint of life itself, the molecular basis of inheritance.
So let's start with a number that is just, well, it's pretty hard to wrap your head around. 2.2 meters. If you could take the DNA from a single one of your cells, just one, and stretch it all the way out, it would be about this long. That's taller than most people. And remember, you've got trillions of cells. It's just incredible. So right away, that brings up a couple of huge questions. First, how in the world do you pack that much stuff into a space you can't even see with your own eyes? And second, how did we even figure out that this specific molecule was the secret to heredity in the first place? Well, to get those answers, we've got to play detective and follow the clues in a pretty fascinating scientific mystery. Our story really kicks off back in the early 20th century. You see, scientists knew that traits were passed down, from parents to their kids, but they were stumped by one of the biggest mysteries of the time. What molecule was actually doing the heavy lifting? What was carrying all of this vital information?
Back then, there were really only two main suspects. In one corner, you had proteins. I mean, they're super complex, they do all sorts of things in the body, they seemed like the obvious choice. And in the other corner was DNA, which honestly most people thought was just this simple, boring, kind of repetitive molecule. The smart money was definitely on protein being the star of the show. But then, in 1928, the case took a wild, unexpected turn. A single, really elegant experiment uncovered the first major clue that pointed all the investigators in a completely new direction. And this big clue came from a British medical officer named Frederick Griffith. He was working with two different versions of a bacteria, Streptococcus pneumoniae. You had the S strain, which had this smooth coat and was totally deadly. And then you had the R strain, which had a rough coat and was completely harmless.
Okay, so Griffith runs a series of tests. Follow me on this. He injects the harmless R strain into a mouse, and the mouse lives. No surprise there. He injects the deadly S strain, the mouse dies. Also totally expected. Now here's where it gets interesting. He takes the deadly S strain, kills it with heat, injects it, and the mouse lives. Makes sense, right? But then came the twist. Griffith mixed the harmless, living R strain with the dead S strain, and the mouse still died.
I mean, this was shocking stuff. It meant that something, some substance, from the dead bad guy bacteria had somehow been transferred to the living good guy bacteria, and it permanently changed them, or transformed them, into the deadly version. Griffith called this mystery stuff the transforming principle. He found the clue, but the identity of the culprit, still a complete mystery.
So Griffith's discovery just kicked off a whole new phase of the investigation. For the next 20 years or so, researchers would build on what he found, which led to a series of experiments that would finally, and I mean definitively, close the case.
This timeline really lays it out for us. You've got Griffith kicking things off in 28. Then in 1944, Avery, McLeod, and McCarty take this huge step forward by figuring out what that transforming principle actually was. And the final undeniable proof came in 1952 from Hershey and Chase.
The team led by Oswald Avery was incredibly methodical. What they did was they took the heat-killed S strain and they treated it with different enzymes. These are basically molecules that can chew up specific substances. So enzymes that destroyed proteins? Didn't stop the transformation. Enzymes that destroyed RNA? Nope, didn't stop it either. But when they used an enzyme that only destroyed DNA, the transformation stopped dead in its tracks. They had found their culprit.
But some scientists were still not totally convinced. So the final nail in the coffin came from an experiment using these little things, bacteriophages. You can think of them as tiny biological syringes. They're these super simple viruses made of just two things, an outer protein coat and an inner core of DNA. The absolute perfect tool to settle this protein versus DNA debate once and for all. So Alfred Hershey and Martha Chase designed this just brilliantly simple experiment. In one batch of these viruses, they put a radioactive tag on the protein coats. In a second batch, they put a different radioactive tag on the DNA core. Then, they just let both batches of viruses infect some bacteria and waited to see which radioactive tag actually made it inside the cell.
And the results? They were just undeniable. After the viruses did their thing and injected their genetic material, only the radioactive phosphorus, the tag that was on the DNA, was found inside the bacterial cells. The protein coats? They were just left outside like empty shells. It was the final definitive proof. DNA, not protein, is the stuff that carries the instructions for life. case closed.
Okay, so the case of the missing genetic material was finally solved. But you know how it is in science? Solving one mystery just opens up a bunch of new ones. If DNA is the blueprint, how does a 2.2-meter-long molecule get packed into a microscopic nucleus? And how is that blueprint copied so perfectly every single time a cell divides?
Well, the solution to the packing problem is a masterpiece of biological engineering. The negatively charged DNA molecule wraps itself around these positively charged proteins called histones. This basic unit is called a nucleosome. It creates the structure that kind of looks like beads on a string, which can then be coiled and coiled again into a super tightly packed chromosome. It's an unbelievably efficient storage system.
But just having a blueprint stored away isn't enough, right? For life to actually go on, for you to grow from one single cell into the person you are today, that blueprint has to be copied, and copied flawlessly, trillions and trillions of times. This incredible process is called DNA replication, and it is fundamental to all life as we know it.
And it all happens through this beautifully elegant process called semi-conservative replication. Now that sounds complicated, but it just means that when DNA is copied, each new double helix that's created is made of one of the original strands and one brand new strand. It's the perfect way to make sure you get an exact copy while still holding on to the original template.
Here's how it works in a nutshell. First, an enzyme called DNA helicase comes in and unwinds, or unzips, the double helix. Second, each of those separated strands now serves as a perfect template. And third, new building blocks. The A's, T's, C's, and G's of the genetic alphabet are matched up to each template, and boom, you have two complete identical DNA molecules right where you started with one.
So we figured out what the blueprint is, we've seen how it's stored, and we know how it's copied. But how does this static code actually, do anything? How does it build and run a living, breathing organism?
This happens through something called the central dogma of molecular biology. Think of it like this. The DNA is the master blueprint, and it's kept super safe inside the nucleus. To get a job done, it gets transcribed into a sort of disposable working copy called RNA. That RNA message then travels out of the nucleus to be translated into the final functional product, a protein.
That very first step, transcription, works a lot like replication. An enzyme called RNA polymerase finds the start here sign on a gene. It then moves along the DNA, reading the code and building a matching copy out of RNA. When it gets to the stop sign at the end of the gene, it just lets go, releasing this new RNA message to go carry out its instructions.
And that really brings us to the most incredible part of this whole story. It all boils down to this. Every single instruction for every trait in every living thing is stored in the specific sequence of just 4 chemical letters: A, T, C, and G. The immense, mind-boggling complexity of life is written in a stunningly simple code.
And that leaves us with one last, really fascinating question to think about. We've cracked the code, sure, but we're really just beginning to read the book of life. If this simple alphabet can write the story of everything, from a tiny bacterium to a giant blue whale, what other profound secrets are still locked away in that sequence, just waiting for us to discover them?